Hi, my name is Angelina Lopez. I am in the microbiology class, and my professors are Professor Caleb Arcora and Sarah Rogers. Professor Sarah Rogers. So today we'll be going over the final exam video, and I did write down the questions just so I remember what is being asked. So I will be going over that. Okay, so the first question says, explain the difference between gram-positive and gram-negative cell walls. So the difference between the gram-positive and gram-negative cell walls is that the gram-positive cell walls are composed largely of peptidoglycan cell layers. It also lacks an outer membrane and stains purple for gram-positive. For gram-negative, it stains pink, and it is kind of the opposite of gram-positive, whereas the gram-negative is thin layers of peptidoglycan cell walls. I'm sorry. Okay, so the second question says, describe the procedure for performing a gram stain. What is a procedure? So first, the procedure of a gram stain is the method of staining that is used to classify the bacterial species into two large groups, gram-negative and gram-positive. And then it says, describe the procedure for performing a gram stain. So the procedure for performing a gram stain starts with applying a primary stain, which is crystal violet. Secondly, you add a mordant, which is the Gram's iodine. After that, what you're going to do is rapid depolarization with ethanol, acetone, or a mixture of both. And then lastly is counterstaining the safranin. So it says, what does each mordant do? So what each mordant does is the crystal violet adheres to the cell membrane, and the second one, which is the applying the mordant, forms coordination complex between the dye. Okay. Third one, which is rapid depolarization. I'm sorry, I'm just looking at the question. Third one, which is rapid depolarization, interacts with lipids of the membranes. And the last one, which is counterstaining the safranin, does not affect the purple color of the gram stain. Now it does stain red though. All right, and the last question says, how is the gram stain used clinically to help identify the cause of infection? All right, so the gram stain is used because it helps us classify the two types of bacteria, gram-negative or gram-positive. And gram-positive, which stains purple, and gram-negative, which stains pink, tells us what type of cell it is, which helps us determine what type of bacteria it is. And once you determine what type of bacteria you have and that you're dealing with, you'll know what to do about it and how to, what antibiotic you'll have to use to get rid of that infection. Thank you.